Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Alina Aisyah and here is my final year project presentation. The title of my research review is The Life Cycle of Sitophilus Species on Rice, a review supervised by Mr. Tun Muhammad Firdaus bin Aziz.
The first part, Introduction.
The presence of insect pests is the common problem for stock products. Numerous techniques have been applied in order to solve this issue, and one of them is the application of methyl bromide. Unfortunately, due to its potential of depleting the atmospheric ozone layer, the usage is regulated as stated by Morrissey 2006. As an alternative, phosphine is used as pest control. However, Naya et al. 2019 stated that multiple insect species have developed phosphine resistance, which is in accordance with the Kim et al. Recent study that claimed that many insect species have reported phosphine resistance in many nations. Insect pest management is an environmental sensitive approach to pest management. According to Biology Online, life cycle is defined as a course of development of an organism, comprising of the whole life history of the organism. A recent study conducted by Singh 2017 stated that a completed life cycle is actually depends on the rice variety on which the insect is thriving. Background of study. Oryza sativa, or also known as rice, has become the main crops in the world. Duna and Roy 2013 pointed out that the rice accounts for 60% of food consumption in Southeast Asia and 35% in East and South Asia. In Malaysia, rice has become the main food and the production of rice is consistent to fulfill the high demand as highlighted by Sharifah et al. 2018. According to Panessa and Co. 2016, there are about 500,000 accession of rice genetic resources conserved across the world, with over 40,000 rice varieties available. This large number of rice varieties are due to application of genetic engineering and gene editing to modify these varieties for specific traits. There are about 11 different colors of rice varieties present. The color of the grain can be an indicative of the nutritional content. Based on previous study by Dunia and Roy, they stated that the length of the width of the grains are different according to its varieties. Grain storage in warehouses is critical for maintaining production amounts and ensuring an adequate supply of food for population. Unfortunately, insect pests have been the major constraint to this sector. Akhtar et al. 2017 mentioned that 70% of these stored pests are from root coleopterans, and genus Sitophilus is one of the most damaging insect of storage insects. So, how do these tiny creatures can cause a lot of problems? The adult weevil infestation can cause a significant reduction of carbohydrate content in rice. Meanwhile, the larvae minimize nutritional value of rice as it removes large percentage of protein and vitamins in germ layer, which can reduce the grain weight and the rice quality. Previous study by Singh 2017 proposed that a completed life cycle is actually depend on rice variety on which the insect is thriving. The life cycle of Sitophilus species are quite similar, which comprise of 4 definite stages, which are egg, larva, pupa, and adult. In this review, the life cycle of the 3 species on rice will be compiled, along with the factors that influence the developmental stages of Sitophilus on rice.
It is a comprehensive compilation of the life cycle of the 3 Sitophilus species, which can fill the gap of information related to their biology on rice. This review also highlights the differences in developmental stages of Sitophilus on rice. The objectives of this review are to differentiate the life cycle of the 3 Sitophilus species on rice and to identify the factors that influence the developmental stages of Sitophilus, to describe the effect of physical and chemical characteristics of rice varieties towards the life cycle of Sitophilus species. Research questions of this review covers on how rice influences the life cycle of Sitophilus, which asks on how physical and nutritional content of rice affect the developmental stages of Sitophilus species. Besides, this review also asks on what factors that influence the life cycle of Sitophilus on rice and how rice varieties can affect the developmental period of Sitophilus. Lastly, why there are differences shown in developmental stages between the 3 Sitophilus species on rice. Moving on to the next part, literature review. The taxonomy of rice started with kingdom of Plantae, class of Liliopsida, family of Graminae, and genus of Oryza. According to Duna and Roy, there are 35 species, but only 2 species are cultivated while others remain as wild. Among the 2 cultivated species, Oryza sativa is the most widely grown according to the Department of Health Australia.
The length of brown rice range from 6.87 to 7.76 mm, width of 2.46 to 2.94 mm, while the length of white rice is 6.38 mm, width of 2.24 mm, and the moisture level is 10.37% for brown rice and 12.7% in white rice. It can be concluded that white rice have low nutritional content due to the milling and polishing process. For black rice, the length is 5.62 mm, width of 2.15 mm, while the length of red rice is 5.13 mm, width of 1.90 mm. The moisture level of black rice is 11.07%, and for red rice can range from 9.3% to 13.1% of moisture level. The labeled dorsal image of Sitophilus species. A unique information on Sitophilus is, according to Singh et al. 2018, the red to yellow spots can be in various sizes on each electron due to temperature. In terms of sexual dimorphism, several previous researchers focused on external parts of Sitophilus, such as rostrum, prothorax, and abdomen. However, Aziz and Kasim 2020 showed disagreement as they claimed that external parameters failed to become a solid proof and cannot be statistically proven. Therefore, dissection is the only way to identify the gender for Sitophilus species.
Sitophilus oryzae are commonly found in temperate zone. Their size range from 2 to 3 mm and reddish-brown in color. The punctuation on their prothorax is irregular shape. Sitophilus granarius can also found in temperate zone with size ranging from 3.1 mm to 4.8 mm. Their color can be reddish-brown with oval cone-shaped punctuation. For Sitophilus zimes, they can be found in tropical areas and can also live in colder climates. Their size can ranging from 2.5 to 4.0 mm with darker body color. They have circular shaped punctuation on their prothorax.
Now, let's focus on the life cycle of Sitophilus species on rice. For number of eggs produced, it can be seen that Sitophilus zimes recorded the highest egg production, which is up to 400 eggs compared to other Sitophilus. In terms of favorable temperature, it can be concluded that they have similar range of temperature of 15 degrees Celsius to 34 degrees Celsius and relative humidity of 58% to 89%. This is in accordance with previous study by Hassan et al. That stated that the rate of egg laying increased as the temperature and relative humidity increased. For egg incubation, AISYAH recorded the longest period, which is 3 to 10 days. For larvae in stars, Ascarius showed the longest period when thriving on rice, which is 21 to 35 days, while zimes recorded 22 days. In terms of period pupa, zimes showed the fastest development, which took 6 to 7 days compared to other Sitophilus. The longevity of the adults on rice, AISYAH recorded 3 to 4 months, which is the shortest lifespan, however able to produce up to 8 generations per year. The average total life cycle of the 3 Sitophilus on rice is 39 days.
Temperature and relative humidity is the first factor that influences the developmental stages of Sitophilus species on rice. This is because egg hatching can be halted due to low relative humidity. Plus, different rice varieties contain different level of moisture content. Second is competition. AISYAH is primary pest and storage product, and based on previous study, the smaller larvae produced by S. granarius need to compete with large larvae from AISYAH and S. zimes. Next is food resources. In terms of physical characteristics, Sitophilus species prefer large grain and polished surface, which is in accordance with Atunes et al. 2016.
It is reported that the nutritional content of rice can influence the developmental stages and growth of Sitophilus based on the recent studies conducted by Asuti. The developmental period of AISYAH recorded fastest in red rice compared to other rice types. The development in white rice showed the longest period due to its low nutritional content. According to Okpa et al. 2021, Sitophilus infest rice to obtain essential nutrients for metabolism. This is in accordance with Ojo and Molay, which stated that the balance of nutrients include protein, starch, fiber, minerals, and vitamins affect the developmental period.
Conclusion and recommendations.
The life cycle of the 3 Sitophilus species on rice are compiled, which can be a base information for future references. It can be concluded that the 3 Sitophilus species share approximately similar range of temperature and relative humidity. Factors influence the developmental stages in life cycle of Sitophilus are temperature and relative humidity, food resources, and competition. Physical characteristic and nutritional content of rice also influence the developmental period of Sitophilus. Unfortunately, this review has limitation as the life cycle of both S. granarius and S. zimes on different rice varieties are limited. To address the limitation of this review, future researchers need to focus on life cycle of S. granarius and S. zimes on different rice types. It will significantly improve the knowledge gap regarding their biology. Next, beside rice, future research also need to emphasize of influence of the other hosts such as maize and wheat to the life cycle. These findings act as a great comparative approaches for generating a completed information on their biology and life cycle. Lastly, research facilities need to utilize natural ingredients for pest control without jeopardizing environmental health due to pesticides.